sought after the MSc program, thanks to the visionary leadership of Professor Radhakrishna Pillai, has been well appreciated. RGCB has tasted all its success within the shortest possible period. The future path of RGCB is promising. A bunch of promises to keep that the world is looking forward to us, but we have miles to go before we sleep. These expectations are on our shoulders. We shall proceed and cross the winning ribbon to make this nation proud. We are celebrating the Foundation Day today, and I am honored to introduce Professor Padmanabhan Balaram to all of you. I have known Professor Balaram for many years, over 3 decades. Professor Balaram is a world-renowned biochemist and former director of Indian Institute of Science, Bengaluru. Professor P. Balaram graduated in chemistry from Pune University, Pune, in 1967, and he joined the Indian Institute of Technology, Kanpur, for his master's in chemistry, 1969. He did his PhD from Carnegie Mellon University, Pittsburgh, in 1972 and underwent postdoctoral training at the Department of Chemistry in Harvard University. Subsequently, he joined as a faculty in Indian Institute of Science. He is the recipient of Padma Shri and Padma Bhushan, followed with Claude Prize and among many other notable awards. He has contributed extensively to the area of molecular biophysics and chemical biology. He is currently associated with National Center for Biological Sciences, Bangalore, as the DST YOS Chair Professor. Professor Balaram has authored more than 500 research publications and is a fellow of all the Indian academies, including the Indian National Science Academy. I am thankful to Professor Balaram for having agreed to a talk on a promising subject, chemistry, biology, and the unity of nature. I hand over to Professor Balaram now. Thank you, Balaram. Professor Balaram.
Thank you very much. I hope all of you can hear me. I'm very grateful to Dr. Chandrabas Narayana for inviting me to give this Foundation Day lecture. I have very fond memories of visiting RGCB in its very early days when Dr. M.R. Das was the first director. And over the years, I have had other occasions to come, and more recently, I have met Dr. Radhakrishna Pillai on many occasions. In giving this Foundation Day lecture, I decided to talk about a subject which might be of some general interest to not only biologists, but also to chemists and even people generally interested in science. I've chosen as my subject the connections between chemistry, biology, and how it helps us in our understanding of nature. On my very first slide, I've pictured one of the foundational pillars of chemistry, that is Mendeleev and his periodic table, and the other is a foundational pillar of biology, that is Darwin and his ideas on the origin of species and the fundamental concept of natural selection. In the middle of the slide, I have a picture, and that is the many images of the coronavirus that you can now see on the internet. And the coronavirus has done one thing. It has reminded us very sharply of how powerful biology can be. In fact, if I may be permitted to say, the pandemic has reminded us of how powerful nature really is. It has also reminded us of how biology can sometimes trump both politicians and religion. Biology is something that we need to understand. And the only way you can understand biology in its ultimate detail is to understand the underlying chemistry of biology. Now, once again, my slides, for some reason, do not move.
Can you click on the slide? Now it's to go.
The second slide tells you a little bit about what I have found in the literature on the evolution of Darwin's ideas on the origin of life. In March of 1863, Darwin, in a letter to a friend, said it is mere rubbish thinking at present of the origin of life. One might as well think of the origin of matter. Here is a very important statement. If we want to think about the origin of life, we must think about the origin of matter and all the chemicals that form matter. So 8 years later, in another letter to the same friend, Darwin said that it is often said that all the conditions for the first production of a living being are now present, which could ever have been present. And then a very important sentence. He said, but if, and oh, what a big if we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. This is in many ways one of the first times in the literature that the word protein has really been used. And when I read this, I was quite excited. What would Darwin have actually been thinking about? At that time, nothing was really known about the structure of proteins. But if you want to know about the structure of proteins, you have to go to biochemistry. And one might ask, when was biochemistry born? Biochemistry's birth can be traced to Buchner's famous experiment on cell-free fermentation by yeast extract. What Buchner did was fairly straightforward. He took yeast extracts, he crushed the cells, and he took the soup which came. The soup now did not contain any cells, but only contained the chemicals derived from the cells. And he used the soup and demonstrated that you could now convert sucrose into alcohol. So the process of fermentation was now achieved. You did not need the living cell, you only needed the chemicals which were inside the living cell. And this was the starting point of biochemistry. In the middle of the 19th century, around the same time that Darwin was thinking about the origins of species and the origin of life, Mendeleev was putting together his periodic table. I show you a truncated version of the modern periodic table so that it will fit into the slide. I haven't gone all the way down, but you can see that I've colored the elements. I've colored, for example, in light green, the elements that are important in organic and in biochemistry. But I've also colored in light pink the other elements which are important in biochemistry, and you can find there are many. The alkali metals, sodium, potassium, the alkaline earths, magnesium, calcium, some of the transition elements, manganese, iron, nickel, cobalt, zinc, all of them. Molybdenum, which is found in the enzyme nitrogenase. And today we even have other elements which have been found in enzymes. For example, tungsten has been found in enzymes, which are involved in very interesting hydration reactions. So nature uses many of the elements which are found in the Earth's crust. And this is how both chemistry and biology must have evolved on early Earth. I would make one request. If you can all turn off your microphones, that would help, because otherwise there's a background. On this slide, I ask another question. How did matter itself form? And you can do no better when you ask that questions than go to a very famous book, The Ascent of Man, by the physicist Jacob Ronowski. Ronowski described in a BBC series in the early 1970s, it was an early television series, The Ascent of Man, and he traced the evolution of technology and the evolution of man. In that, he asked some questions. And he says this, in all the stars, there are going on processes to build up the atoms, one by one into more complex structures. Matter itself evolves. He then adds, the word comes from Darwinian biology, but it is the word that changed physics in my lifetime. He describes in poetic terms the formation of carbon in stars, because it's in stars that all the elements are formed. And he says, carbon, for instance, is formed in a star whenever 3 helium nuclei collide at one spot within less than 1/1,000,000th of a millionth of a second. Very, very short time scale. Every carbon atom in every living creature has been formed by such a wildly improbable collision. If you think about this sentence very carefully, you will come to the conclusion that life is extremely improbable. And in fact, life is improbable because we haven't found life yet on the many, many planets which are outside the Earth. In our solar system, nearby planets, and so on. There is no evidence so far clearly found for the evidence of life anywhere else. Biology is very important, and so are Darwin's ideas. And one of the most famous evolutionists of the 20th century, Ronowski, said this a long time ago, nearly 50 years ago, in an article in the American Biology Teacher. The title of the article is quoted repeatedly. He said, nothing in biology makes sense except in the light of evolution. Now, you are in the Rajiv Gandhi Center for Biotechnology. By using the word biotechnology, you imagine that all our understanding of biology can now generate useful technologies. But our understanding of biology even today is quite imperfect. Dobzhansky asked a very important question. Does the evolutionary doctrine clash with religious faith? And then he answered, it does not. It is a blunder to mistake the Holy Scriptures for elementary textbooks of astronomy, geology, biology, and anthropology. Only if symbols are construed to mean what they are not intended to mean can there arise imaginary, insoluble conflict. He wrote this sentence in the 1970s because the constant conflict between Christianity and evolution, biological evolution, is often challenged by those who are religious fundamentalists. But it turns out that biological evolution explains much of nature as we see it around us. But in order to understand nature, the most important subject is chemistry. Arthur Kornberg, the man who discovered DNA polymerase and fired the first shot of the biotechnology revolution in the 1950s, many years later, in the late 1990s, when he was old, he called chemistry the lingua franca of the medical and biological sciences. One of the saddest parts of Indian education is that very often, neither in medical education nor in biological education is the importance of chemistry really stressed. But it turns out that in order to make important advances in biology today, it is necessary that one has a very strong chemical foundation. We are now in the year 2020. 2 decades of the 21st century have gone by. But in the late 1990s, when we approached the year 2000, it was very popular to look back on the century, the 20th century, and also to look back on the millennium and ask some questions. The journal Nature at that time commissioned a set of essays, one-page essays which were easy to read, and every week somebody would write about what they considered was the most important scientific advance of the 20th century. Every one of you who's listening to this, you have your own ideas of what was the most important scientific advance of the 20th century. I was then the editor of Current Science, and every fortnight I would have to write an editorial. And so I would avidly read these essays in Nature. But the one essay which has remained with me after 20 years is the essay that I picture on this slide. This was an essay by Václav Smil, a Canadian scientist, who wrote an essay entitled Detonator of the Population Explosion. And this sentence that he wrote really stayed in my mind. He said that the world might be better off without Microsoft and CNN, and nuclear reactors nor space shuttles are critical to human well-being. But the world population would not have grown from 1.6 billion to today's 6 billion. Today, 20 years later, we are much past 7 billion with the Haber-Bosch process. What is the Haber-Bosch process? It's the industrial synthesis of ammonia, which I picture on the next slide. And it is this synthesis that really detonated the population explosion. How did this happen? Because it was only after ammonia was synthesized industrially that urea could be made. And it was only when urea was used as a fertilizer that famines disappeared and death due to famines also came down dramatically. The result of this is that chemistry has contributed more than any other subject, really, to this advance in the well-being of human populations. Nature itself fixes ammonia. It fixes nitrogen by taking gaseous nitrogen from the air and then reducing it with hydrogen to give you ammonia. But this is done at room temperature, under normal conditions, in aqueous solution, but you use this remarkable enzyme, nitrogenase. This is a central reaction of biology, the nitrogenase reaction. But you can now see the connection. The connections are now becoming visible. A great deal of the chemistry that we do helps us to understand biology. At the same time, a great deal of the chemistry that is done takes us forward technologically. The second chemical, which I would like to highlight here, is DDT, dichloro-diphenyl trichloroethane. DDT appeared in the mid-1940s, just as the Second World War was winding down. It was a remarkable chemical because what it did was it killed insects. And therefore, you could kill insects in the field. You could also kill insects which caused human disease, like you could kill the malarial mosquito. Here it was a one simple molecule which had immense applications in both agriculture and medicine. All of you are in an institute for biotechnology. Can you imagine making an impact like the discovery of DDT? This was recognized by a Nobel Prize almost immediately. The award went to Paul Muller. But many years after DDT was used, it became clear that DDT has a great deal of side effects. And Rachel Carson wrote this book in 1962, Silent Spring. At that time, people were appalled by what had happened at Hiroshima and Nagasaki when the atomic bombs had been exploded. So she says, we are rightly appalled by the genetic effects of radiation. How then can we be indifferent to the same effect in chemicals that we disseminate widely in our environment? In your own state of Kerala, there has been this continuing controversy on endosulfan usage. And therefore, one must think about the importance of the environment and what happens when we disseminate chemicals into the environment. But Rachel Carson's 1962 book is the start of the environmental movement. Today, it is very popular to be an activist and an environmentalist. But if you look back at history, you will see that Rachel Carson's book is really the first shot of the environmental movement. But chemistry and biology are intertwined in other ways. Here is an example. This is from an article which appeared in the Proceedings of the National Academy of Sciences a few years ago, written by Tom Eisner, who started the field of chemical ecology. And he says this, new disciplines arise by a convergence of interest. Chemical ecology is the product of a partnership between biologists and natural product chemists, united by shared vision and empowered by complementary skills. I like this very much because he talks about a shared vision and being empowered by complementary skills. This is what is needed for our institutions to move forward, where people have a shared vision and they all bring to the table different skills and are collaborative. What was Eisner's vision? Eisner's vision stems from the realization that all organisms emit chemical signals and that all in their respective ways respond to the chemical emissions of others. This is true. Human beings are the only animals who really communicate by sound and by language. Every other biological organism from microorganisms to insects, plants, and so on. All of them communicate with their environments exclusively by means of chemicals. Chemical communication in biology is why are there so much why is there so much chemical diversity in biology? What are all these chemicals now used for? This understanding of the relationship between chemistry and biology, I think, is central to all Indian institutions which work in subjects which lie at the borders of chemistry and biology. In the year 2000, I read another article, this by the famous molecular biologist Sidney Brenner. Sidney Brenner was an acerbic critic of everything that was going on in science at that time. And he said this. He began an article very provocatively saying, I once made the remark that two things disappeared in 1990. One was communism, the other was biochemistry, and that only one of these should be allowed to come back. He, of course, I don't think had visited Kerala at that time. But both communism and biochemistry are alive and well. He did recognize that biochemistry has to be there, and he went on to say we do not have to resurrect biochemistry, and it will flourish because it provides the only experimental basis for the causal understanding of biological mechanisms. Why did he say this? This is because in the sweep of molecular biology, what one forgot classical biochemistry and chemistry. To most students of biotechnology today, what they deal with are faceless reagents which appear in kits. Very often they do not know even what those reagents are and what those reagents do. All they know is that if they follow a certain protocol, something will happen according to what they want. Biochemistry, even in its simplest form, can be very powerful. I illustrate this with the other Nobel Prize of the 1940s, this one given immediately after the Second World War. An extraordinarily practical problem. You might call this biotechnology. The Nobel citation said it was given to Arturi Virtanen, a Finnish scientist, for his research and inventions in agricultural and nutritional chemistry, especially for his fodder preservation method. Fodder needs to be preserved, but fodder simply rots. And in the long Scandinavian winters, if you want to preserve fodder, what you need to do is discover methods by which preservation can happen. Virtanen in the 1920s systematically showed that harmful fermentations can be stopped by lowering the pH. And the best way of doing this was to add a mixture of sulfuric and hydrochloric acids. Today, of course, we've understood that if you want to stop undesirable organisms from growing, you must add other organisms, lactic acid producing bacteria, for example, and certain other preservatives in silage. But this was an agricultural advance worthy of a Nobel Prize. But at its heart was an understanding of the biochemistry of fermentation and how this biochemistry could be manipulated for a useful purpose. Having shown you the importance of both chemistry and elementary biochemistry, I want to tell you about the three pillars of modern biology. Biology today rests on three pillars. The first of these is evolution, the ideas of evolution. You're not a biologist if you have not studied evolution and tried to understand in your own way what it means, what does natural selection and what does random variation really mean. The other pillar was also erected in the middle of the 19th century by Gregor Mendel, and that's the idea of genetics. How are hereditary characteristics passed down from generation to generation in biology? But what is the link between genetics and evolution? It is chemistry. That chemistry is the understanding by Oswald Avery in the 1940s that DNA is a genetic material. And the finding of Watson and Crick that you could now derive a structure for DNA based on X-ray diffraction data which told you how DNA could be multiplied and how could information be transferred from generation to generation. But if you want to understand biology, what do you want to understand? You want to understand nature. You want to understand all the organisms in nature and their behavior. For example, if you want to understand how does the female silk moth attract the male silk moth in order to mate, it must release a pheromone. You need to understand the structure of the pheromone, the biosynthesis of the pheromone. How is the pheromone recognized by a biological receptor, and eventually signal transduction and many of the other important modern areas of molecular and cellular biology. But in order to do all of this, how does all of this evolve? It evolves over time by variation, by random variation in genes and selection. There are two dominant technologies of our times, information technology and biotechnology. This slide is just to remind you that both these technologies have their roots in fundamental science. Today, when I lecture or listen and sit in meetings which discuss biotechnology, I feel that there is a general feeling that fundamental science has done whatever it can and that we need to somehow move to translational research very quickly. But it is important that we do not neglect fundamental science. Claude Shannon and Oswald Avery are the two iconic figures whom you might call the founding fathers of information technology and biotechnology. Because if Avery had not invented DNA, you wouldn't be talking about, had not found DNA, you wouldn't be talking about biotechnology today. Claude Shannon, interestingly, there's a connection. Both of these were done in the mid-1940s. And Shannon's PhD dissertation is entitled An Algebra for Theoretical Genetics. So he could already see some sort of connection. Today, when we talk about genomic information, information technology is most important in handling this information. If we talk about artificial intelligence, which has now again come from many advances in computer science and information technology, it turns out that where we would be applying artificial intelligence most effectively may be in areas of biology and medicine. But let me come back to biochemistry. Mendeleev's periodic table hangs in every chemistry laboratory and nobody looks at it. The metabolic chart also hangs in every biochemistry laboratory and nobody looks at it. Why do we not look carefully at the metabolic chart? It's because it's terribly complicated. It's full of chemistry and biochemistry, chemicals being converted into one another. But all of these processes need to happen, and they need to happen precisely and without failing in order for a cell to function. And if sometimes a particular reaction fails, there are alternative pathways which the cell then uses. So like an electrical network, there is a great deal of redundancy built into a biochemical network. And that's what the metabolic chart really signifies. Today, there are people who are trying to understand the metabolic chart and understand what one might call the logic of metabolism. Because you must understand the logic of metabolism because you have to ask the question, how did life evolve on Earth? How did all of this complex biochemistry really come about? And just for the students who might be listening, I want to tell you that this is a modern area of thinking. In an article in Chemical Reviews, Walsh and his collaborators now ask, what are the most important molecules for biochemistry? Suppose you were asked to say that there must be only 8 molecules that you must pick. Which would be those 8 molecules? You must go to this article to see what those 8 molecules are. The 3 major pathways which everybody studies, glycolysis, the citric acid cycle, and mitochondrial oxidative phosphorylation are all illustrated on this slide, and the interconnections between them. This is energy metabolism in a cell, because after all, life depends on energy. But let me come back to biochemical pathways. Pepper is very popular, and so is the red chili. And then you ask the question, what is the pungent component of the red chili? It is the molecule capsaicin, which is pictured on the right-hand side of the slide. A very simple organic structure. How does the plant really make this? What is on the left are the biochemical pathways by which it makes it. And it makes it starting with two simple starting materials, the amino acid phenylalanine on the left and the amino acid valine on the right. So amino acid biosynthesis, then you have these amino acids, and then you go forward. But look at the number of chemical steps that need to happen before you come to capsaicin. It's a remarkable number. And on each chemical step, the arrow, there is an enzyme which is indicated. That enzyme, phenylalanine ammonia lyase, cinnamic acid 4-hydroxylase, these are the enzymes which catalyze those chemical reactions. The reactions will not happen by themselves. They're too slow. For biochemistry to work, the reaction must be catalyzed, and so there must be an enzyme. Today we look upon enzymes, sometimes we're so familiar with enzymes that we look upon them with contempt. But you should never do that. Those enzymes need to be made by the cell. How do enzymes get made? They need a gene. And if they need a gene, then every enzyme, many genes are important in that biosynthetic pathway. But I'm going to digress for a moment to tell you the importance of genes and the importance of enzymes. And I'm going to do this by taking an article that I read early this year. I read this before the pandemic, and very interesting title, appeared in the New Yorker magazine. Does hurting make us human? You know, we hurt, we feel pain. That is a human characteristic. But here there is this Scottish lady, Joanne Cameron, pictured on the slide, who didn't seem to feel any pain. She didn't feel pain. And what was most interesting was her negative emotional range is limited. How nice it would be if all our negative emotional ranges were limited. And especially if the negative emotional ranges of politicians were limited, then life would be so wonderful. Joanne Cameron went for surgery and told the anesthetist that she didn't need anesthesia. He didn't believe her. He still gave her anesthesia. It was a very painful orthopedic surgery. But the next morning when he came back to check up on her, the nurse said that she had refused any analgesic and she had taken only one paracetamol. He was really surprised. Further investigations were done, and then they found that in addition to an insensitivity to pain, she also never felt unhappy. Even when her mother had died, she somehow converted that into looking on all the wonderful aspects of her mother's life. So her brain obviously operated differently from your brain and mine. Why was this? This is the paper which then appeared in the British Journal of Anesthesia in 2019. And you can see the title, Microdeletion in a FAAH, that is fatty acid amide hydrolase, an enzyme, pseudogene identified in a patient with high anandamide concentrations and pain insensitivity. So there's a molecule, anandamide, which has been introduced there. And I show you the chemical structures of anandamide. Anandamide on the left, and the enzyme fatty acid amide hydrolase breaks it down into arachidonic acid and ethanolamine. In our own biochemistry, both arachidonic acid and ethanolamine are commonly found. Arachidonic acid is a product of lipid metabolism. Ethanolamine is a constituent of the neurotransmitter acetylcholine and also the phospholipid phosphatidylethanolamin. These two combine in a biosynthetic process to give you the molecule anandamide. But anandamide must be broken down. If anandamide does not break down, you have the kind of phenotype or the kind of sensations that the Scottish lady really had. What is anandamide? Anandamide is a molecule which binds to the cannabis receptor, the cannabinoid receptor. Today in our newspapers, marijuana is very much in the news because every time a film star is found with a little bit of marijuana, they are arrested and the Narcotics Bureau is after them. But marijuana has been used, hashish, bhang, these have been used in India for thousands of years. And the active constituent of marijuana is tetrahydrocannabinol, which is on the top left of the slide. The structure was determined in 1964 in the Journal of the American Chemical Society. Anandamide is the molecule at the bottom. It's found in brain, and it was isolated from brain in the 1990s in a paper in Science. Both these pieces of work were done by one scientist, Raphael Mechoulam, working in Israel. He is today called Mr. Marijuana, and this is what he did. He had determined the structure of tetrahydrocannabinol in the 1960s and asked the question, how can a molecule made by a plant have a profound effect on your brain? There must be something in your brain which recognizes that molecule. Therefore, you must have an endogenous molecule which binds to the same receptor. So he then took pig brain, 4.5 kilograms of pig brain, and isolated from it less than a milligram of anandamide and determined its structure. This slide tells you the confluence of chemistry and biology. Left side is a competitive binding assay using a radio-labeled cannabinol. On the right side is a mass spectrum from which to determine the structure of anandamide. The term anandamide was coined from Sanskrit word ananda, which means bliss. But when Mechoulam was interviewed, he was asked, why did you not use a Hebrew name? He said, we looked for a Hebrew name, but as you may well be aware, Jews are not very happy. We have a lot of words for being down and so on, but not so many words for extreme joy. This is really a reflection of about 3,000 years of Jewish history. But coming back, if you have a substrate and it needs to be converted to a product in a biosynthetic pathway, you need an enzyme. If you need an enzyme, you need a gene. Therefore, for any multi-step pathway, you need many genes. And therefore, for all this kind of secondary metabolite biosynthesis, those genes must be arranged in clusters. And they must be modular assembly with start and stop signals which are unique. The enzymes are very important. But to stress this point, because many of you are molecular biologists, and I know that molecular biologists generally look with disdain upon enzymes, I must quote the high priest of molecular biology, Francis Crick. As early as 1958, he said, the main function of proteins is to act as enzymes. He also recognized something which is remarkable. He said it is at first sight paradoxical that it is probably easier for an organism to produce a new protein than to produce a new small molecule. And then he added at the end, there seems little point in genes doing anything else but protein synthesis. Today we've moved very fast, and many of you will be more familiar than I am with this. Today, if you take eukaryotic organisms, when I made this slide, there were as many as 789 genomes. There are probably a few more now. Human genome, formal announcement was 20 years ago. And you might ask yourself the question, how much has it benefited us to know the complete human genome? How much of current medical advance is based on the knowledge of the human genome? But I put up this tree just to tell you that what I mean by the unity of biology. To get these genomes, you can now build evolutionary trees. And when you build evolutionary trees, you now see that human beings are pretty close to chimpanzees. They're not too far from rats and mice, and they're not too far from pig, cattle, and sheep. In India, there is so much of discussion on pigs, on cattle, on sheep, human beings, and so on. But when you look at slides like these, which tell you how closely we are genetically related, then you will see that in biology, there's a great deal of unity. Today's information technology allows you to look at the growing volume of data in real time. And I just show you one example. The genome size and protein coding genes are correlated. And you can see how data is growing. We are still discussing setting up a national data center. But it will turn out that data has already gone well beyond anything that can be analyzed easily. But in the last part of my talk, what I'd like to do is really to ask a question about the timetable of evolution. And I take the article recent article appeared about 2 years ago in Science Advances. In order to look at the timetable of evolution, we need to have some understanding of the timescales of geology. When did the Earth really form? The Earth formed maybe 567 million years ago. That's where it starts. 5.5 billion years. And now you can now look at the various geological periods which have then been there. When did life first originate on Earth? Probably somewhere about 4 billion years ago. That would have been during what the geoscientists or the earth scientists call the Archean period. The archaebacteria are named after this. But at that time, the earth did not have any oxygen, and therefore everything grew in an anaerobic atmosphere. Oxygen came with the great oxygenation event somewhere maybe around 2,200 million years ago. And that is right in the middle of the yellow bar that you see as the Proterozoic period and labeled with the abbreviation GOE. That's when the first photosynthetic organisms evolved, and they then began to oxygenate the atmosphere. And it was through the splitting of water that the Earth's atmosphere got oxygenated, and then organisms which used oxygen then began to evolve. When you look at this, plants came very late. Animals with bilateral symmetry, bilaterians, came much earlier, and plants colonized land much later. And then, of course, flowering plants came much later. By that time, the larger animals had come. But human beings and their ancestors, immediate ancestors, the hominins, came very much later. You can see they're right towards the bottom end of this figure here in the purple patch at the end. So you can see that we are very, very recent in the grand scale of geology and biology. So the timetable of evolution is summarized on this figure. Two important things: the grand oxygenation event, and then there are other oxygenation events which have taken place to give us our present atmosphere. But today we learn a great deal about all of this from subjects which you would not consider as biology, paleobiology, and a lot of geochemistry, geophysics, isotope dating, isotope geochemistry, which goes into all of this. There is one experiment, an old experiment, which I want to highlight here. This is Anandromide's Origins of Life experiment. He asked the question, what will happen if you take a mixture of simple gases, methane, ammonia, hydrogen, and steam, put them in a glass bulb, and pass a spark discharge through them and collect the residue? He collected the residue, a dark residue, at the bottom of a flask and then analyzed it. When he analyzed it with a paper chromatogram, he saw the chromatogram that you see on this slide. That chromatogram now showed you the presence of the amino acids alanine and glycine. The molecules of biochemistry had now been formed from simple gases under spark discharge. So at the time when this paper appeared in 1953, this was called the Origins of Life experiment by the New York Times. The Watson-Crick paper appeared the same year in Nature, but the New York Times did not report on it at that time because nobody understood the importance of the Watson-Crick factor immediately other than scientists. The Miller experiment captured the imagination because we always wonder, how did life evolve? What would have been the first life forms and so on? But what is remarkable is the tribute to the American habit of preserving everything. Miller's flask had been preserved at the University of Chicago. And maybe 50 years later, when it was reanalyzed by one of his old students with modern analytical chemistry techniques, many more molecules were found. So our ability to find things depends really on the techniques that we have to look at the complexities of the chemistry in biology and the chemistry in nature. That's about simple chemicals and going to more complex chemicals. How do you go from simple cells to more complex cells? What is the last universal common ancestor from which the tree of life diverges? Bacteria, archaea, and the eukarya. Today, there is a lot of work I refer you to a paper in Nature in January 2020, where they have isolated an archaeon at the prokaryote-eukaryote interface. It's a remarkable piece of work because it has taken a dozen years to actually culture this organism, which is an extremely slow-growing organism. But archaea and bacteria may have fused together to give the first eukaryotic cell. These kind of endosymbiotic theories need to be confirmed by experimental findings, and that is what people try to do today. So from simple chemicals, how do you get complex chemicals? From simple cells, how do you get more complex cells? We still don't know how we go from a mixture of chemicals to a simple cell. That we do not know. That is still a complete black box. What do we know about prebiotic chemistry? Prebiotic chemistry, for example, starting with hydrogen cyanide, can give you many, many molecules. Hydrogen cyanide is a wonderful precursor. If you throw in a few other simple molecules, as I picture on the slide, you can get the DNA basis from hydrogen cyanide. This illustrates one of the paradoxes of chemistry. Hydrogen cyanide is the molecule with which Hitler killed millions of Jews in the gas chambers at Auschwitz and other concentration camps. Hydrogen cyanide is also the precursor molecule when we look at prebiotic chemistry. But molecules might have come on early Earth from other places. And here, for example, today, astrophysicists can tell you what kind of molecules are there in the comets. And spectroscopically measured concentrations in the Hale-Bopp comet are what's shown on this table over here. And you can see formaldehyde and hydrogen cyanide are present there. And this is a very recent paper a few years ago, where if you take hydrogen cyanide, hydrogen sulfide, copper, and light, you can make many more molecules. Now you can see the beginnings of metabolism in test tube. So scientists are making great progress in understanding how chemical complexity of biochemistry really evolved. But the question we really are trying to ask is, why is life the way it is? Bacteria evolved into complex life just once in 4 billion years of life on Earth. All complex life shares many strange properties, from sex to aging and death. If life evolved on other planets, would it be the same or completely different? The question which one likes to ask, but very often people do not ask this. For the last part of my talk, I'm going to show you something that I've picked off the internet. Here's the world, populated in 200,000 BC. No human beings at all, except in that little gray shaded area in East Africa. And so that's the current theory. Man originated and moved to other parts of the world with the first Homo sapiens appearing in East Africa. By 32,000 BC, moved all over the world. I want you to look at these years very carefully. 32,000 years BC, approximately 34,000 years ago. So this is our time scale. Time scales are very important in geology, in biology, in chemistry, and in physics. 2,000 years ago, this is what the Earth looked like. By now, human beings had colonized very large parts of the globe. Civilizations had begun to develop. You can see India, China, Persia, the Mediterranean regions, the Middle East, and so on. Almost nothing in the Americas 2,000 years ago, almost nothing in Australia, nothing in Russia, very little in South America. So from this point onwards, the question you might ask is, how do we connect human beings? What is the unity amongst human beings? And today it turns out that ancient DNA might actually rewrite the prehistory of every country and the prehistory of India in particular. Tony Joseph in this book, Early Indians, emphasizes this point. And he says, the new field of population genetics, thanks to an analysis of DNA extracted from individuals who lived tens of thousands of years ago, might tell us something about where we originated from. The ancient DNA analysis is very recent. I retired in 2014. At that time, there was almost no ancient DNA analysis done, very few samples, a handful. Today, you can see it is growing exponentially. And our methods of extracting DNA and of even finding fossils have improved dramatically. and collaborations between paleontologists and biologists have grown. They've grown in the world. They have not grown in India. What is the connection between history and biology? I'm coming to the end. Because when we ask about evolution, we are talking about our evolutionary history. But we have our own understanding of the term history. Yuval Harari, the Israeli historian, wrote this book, Sapiens. If you haven't read it, you should read it. Harari, as a historian, has a better understanding of biology than many biologists. He says that the immense diversity of imagined realities that Sapiens invented and the resulting diversity of behavior patterns are the main components of what we call culture. Once cultures appear, they never cease to change and develop. These unstoppable alterations are what we call history. So our history is cultural history, not biological history. And he says the cognitive revolution is accordingly the point when history declared its independence from biology. Is when the human brain sort of set human beings apart from animals, when we moved from the hunter-gatherer stage and we moved to fixed societies. That is when this happened. What was that point in historical time? That was with the invention of agriculture. The invention of agriculture is what moved human beings from societies which were hunter-gatherers, just like animals, driven by biological instincts, to stationary societies where they needed to protect their fields. In order to protect the fields, they needed to form groups, and they needed to form groups and protect themselves from other groups. And then he says, to understand the rise of Christianity or the French Revolution, it is not enough to comprehend the interactions of genes, hormones, and organisms. It is necessary to take into account the interactions of ideas, images, and fantasies as well. And this is important whenever there is a clash between the history that we are talking about. The history that we are talking about is 3,000 years, 4,000 years, 5,000 years, not more than that. The history that we are talking about in biological evolution or in geological timescales is grand. It's very much more than that. I'll go back to Ronowski. And Ronowski says that among the multitude of animals which scamper, fly, burrow, and swim around us, man is the only one who is not locked into his environment. His imagination, his reason, his emotional subtlety and toughness make it possible for him not to accept the environment but to change it. And that series of inventions by which man from age to age has remade his environment is a different kind of environment, not biological, but cultural evolution. I call that brilliant sequence of cultural peaks the ascent of man. If you go back and read the ascent of man, you will see that every chapter is based on a scientific and technological advance. So science has really contributed to the evolution of human culture. It has contributed to the evolution of human culture in every way, but we do not think about this connection at all. For example, if you have a Ministry of Culture, it is separate from a Ministry of Science. But really, you can ask, how did cultures evolve? They evolved because of technological advances, as he says, from age to age. And that is what has happened. Industrial Revolution more recently, but every revolution in the past, from the Stone Age onwards. Why is it important to understand this? It is important to understand this if you want to understand nature. Ronowski quotes Wordsworth, who said, "For nature to me was all in all." Lastly, cultural evolution is a subject also today of analysis. Papers on cultural evolutionary theory even appear in the proceedings of the National Academy of Sciences in the United States. And here the authors have asked a question, how culture evolves and why it matters. But one question which I'd like to pose to you so that you think about it is, can cultural evolution influence biological evolution? Cultural evolution happens on much shorter timescales, even within a generation. Because, for example, within our own generation, we've seen so many technological advances, social media, information technology, computers, and so on. All these change cultures, the cell phone. But cultural traits can alter the selection pressures on genetic traits over long periods of time. And here is an example. For much of human history, there was little reason to digest milk, because once babies were weaned from the mother's breast, there was little reason for them to digest milk. So the lactase gene, which converts lactose to glucose and galactose, gene was just switched off. The same way that the fetal hemoglobin gene is switched off immediately after birth. But once dairy farming came along and cattle domestication happened, milk consumption began. So now you began to drink milk. And the only people who drank milk and digested it easily were those who had lactase persistence. That is, the lactase gene had not been turned off. So that mutation allowed lactase persistence, and lactase persistence has been selected for over the last 20,000 years or so, since dairy farming began, maybe 10,000 years, over the generations. And it turns out that today, if you look at lactase persistence, the circles that you see on the map of the world, the red parts or the orange parts, they are those regions where there is a lot of lactase persistence, that is, they can digest milk. Those which have the brown or the yellow, those are regions where they don't really digest milk very well. So you can see Scandinavia downwards as we come through Europe. By the time you come to South India, you come down to the south of Africa, you don't have lactase persistence at all. And milk is not an important product. Now, is this important to understand? It is, because tomorrow, if you wanted to do nutritional interventions, say the World Health Organization or the United Nations wanted to nutritionally intervene, it's not a good idea to push milk products down babies and infants and children in Africa. You must look for other things. I've come to the end. I want to say a little bit about the connection between biochemistry, chemistry, and biology, which I think is very important. When I was the director of the Indian Institute of Science, we celebrated 100 years. And in our centenary, one of my ambitions was to get the biologist Carl Lewis to come to India and give a talk. He's the man who discovered the third branch of the tree of life, the archaea. And he wrote back saying that he was old and he was no longer able to travel. Shortly afterwards, he passed away. On the internet, I found this wonderful tribute to him. In this tribute, the quote is attributed to the Greek Pliny the Elder. And what the quote says is this: nature is to be found in her entirety nowhere more than in her smallest creatures. That is, in bacteria, in microbiology. I would like to paraphrase this to say that chemistry is to be found in her entirety nowhere more than in nature's smallest creatures. That is, in microbiology. Because it is only microorganisms which are subject to the harshest selection pressures that are possible. In order to meet the challenges of the environment, they now devise chemical stratagems which allow them to survive in these inhospitable environments. And therefore, when you go in to understand the chemistry, you find entirely new chemistry. That's where I think one of the most fertile fields of research will be there in the future. Lastly, why do I draw your attention to such a disparate group of subjects? It is because of the environment, climatic, social, and political, in which we live today. Some reflection on the roles of chemistry and biology in shaping nature may allow us to introspect on the course of human history and attempt to understand why the world today is the way it is. Science is a deeply humbling subject. And on this foundation day of your institution, I hope I have conveyed to you how any attempt to understand even the smallest of nature's creations can be deeply humbling. Lastly, I turn to the coronavirus, which is a public health nightmare and an artist's delight. How do we define a virus? There is this wonderful definition, which I found in a New York Times report. I traced the origins of this quotation to Peter Medabar's book From Aristotle to Zeus. He calls a virus a piece of bad news wrapped up in a protein. And that's what the coronavirus is. RNA now wrapped up in protein. The spike protein is what you see on its surface. All these wonderful images of the coronavirus. Does the coronavirus belong to chemistry or does the coronavirus belong to biology? Viruses inhabit that what I would call no man's land between chemistry and biology. The line of actual control between biology and chemistry changes every day. And I think it changes much, much faster than the line of actual control between, say, India and China. But today we must realize that the borders between chemistry and biology are completely fluid. And we must go on both directions in order to understand, for example, the coronavirus in its ultimate detail. As a virus, it has no existence by itself. But many viruses are crystalline and their structures can be determined. One day I suspect it will happen to the coronavirus too. But they need to get into a cell, a living cell, before they can reproduce. But then they have the property, they can use the machinery to reproduce themselves. That is what has led to the pandemic. But both chemistry and biology are necessary to understand the coronavirus. I'd like to thank the RGCB for inviting me to give this Foundation Day lecture, to wish all of you the very best. And in my very last slide, acknowledge the two institutions where I have spent almost my entire career. The Indian Institute of Science on the top left, where I spent over 41 years, and the National Center of Biological Sciences, which has provided me a post-retirement home in the last few years, and where my understanding of biology has really improved. Thank you very much for your patience.
Thank you, Professor Balaram. Thank you very much. Can I have the Balaram's video? Professor Balaram, I hope you are able to see us.
I can see, let me just try to see you slightly bigger. Yes, I can see you. I can see lots of people on small
Okay.
boxes. Yeah.
Okay. Yeah. So thank you very much, Professor Balaram. It was a wonderful lecture. I think we have a huge audience, and maybe they have a lot of questions, but I think we'll limit it to about 10 minutes or so, or 15 minutes maximum, depending on how much you can take these questions. So can I ask them to ask you questions?
Sure.
Yeah.
I'm here.
So, yeah. Go ahead, sir. Anyone from here in the audience want to ask any questions? I also open it up for people online if they want to ask questions to Professor Balaram before we conclude the session. You can unmute and ask questions.
Hi, sir. Hi, sir. I'm Jiva. I have a question. Can you hear me, sir?
Yeah, I can hear you. Quick.
Sir, in one of the slide, you mentioned about the lactase production in the humans because of our cultural habit or food habit. Same way, if say the people called vegans who don't consume any animal products have the problem of vitamin B12 deficiency. So in a long time point, if those vegans say for 100 years or 200 years, so the people who don't consume meat and animal products, will they have the ability, I mean any mutation may occur in the, for B12 production in their own system?
I don't think so, because B12 biosynthesis is very complex. Okay, and I don't if you have things which are simple, one enzyme related, it is easy to see how a mutation might help. But if you have a complex biosynthesis, and B12 is terribly complex, it's very unlikely. It's only the microorganisms which can get B12 so easily. But we will get our microorganisms from somewhere else. The vegans only think that they are what they don't see doesn't hurt them.
Okay, sir. Thank you, sir.
You can come. There is a question from audience in our Ramardas hall.
Do you
Yes.
think new viruses are generated as time passes by?
Can you just say that again?
Do you think new viruses are generated as time passes by?
Many viruses mutate fairly fast. It's not that new viruses need to be generated. There are enough viruses already in nature. In fact, if you look at the sea has an enormous number of viruses and millions of them. So one wonders about jumping from marine organisms to terrestrial organisms to man. So I think viruses are there already around us, enough number without our having to generate new ones. But they do change over time, yes.
Lovely. There's one more question here.
I have two questions.
Yeah, okay.
Hi, sir. Sir, I have one basic question. So, after having this much of basic understanding in chemistry and evolution, why we still find difficulty in artificially constructing a cell, or like now there are many organoids which is being made, but when we talk about having life or about that process is still limiting to us.
Yes, it will be limiting for a very long time to come, because you are not going to have what one would call truly synthetic life. No. That is not going to happen simply because of the complexities of biochemistry. Craig Venter actually synthesized a complete viral genome, and then, but then you then need a cell into which you can put that in order for it to make new viruses. But that's not truly complete synthesis. No, we don't understand how. There is a huge gap between our understanding of how chemicals might have evolved and how organisms once formed might have evolved. So how did they form in the first place? There is a gap, a big gulf which needs to be bridged between chemical evolution and biological
Okay, so another question is actually it's from a student side. Basically, those who even understand the whole periodic table, they find difficulty in understanding the basic concepts of pH, concentration, and in my experience, even in lab, students find very difficulty to follow such very basic things, and which I feel which is most crucial in majority of experiments. Even in a minor change in pH, how our results may vary and all those things. So where do you think this shortcoming lies in students? Why they find a difficulty in catching the right point?
The problem is not with the students. The problem is with our entire educational system, which is with teachers and which curricula which have been formed. It may not be a good idea for me to say this over here, but biotechnology has done more damage to biological research in India than any other subject. And this is because we've had courses for now pretty close to 35 years, biotechnology courses where the courses have been designed in such a way that many of the things that students should learn are not there. In many institutions, they're just not taught. And excessive emphasis on techniques rather than on concepts is dangerous. And therefore, you many times use techniques without any understanding of what is behind the technique that you're using. And this is going to be more and more as we go along. This problem is not there in the United States, for example, where the method of teaching and the subjects that people take are different. Here, for example, you can take a 3-year BSc course in biotechnology and a 2-year MSc course in biotechnology without learning many things in those 5 years. It's probably more important to learn a little bit of physics and a little bit of chemistry than it is to learn many of the other things which you can learn later. See, some of us have had a great advantage. We didn't learn any science at all. So we learned it later, but today, right from 12th class, you learn it, you read the same things over and over again. I think we should go back and look at our teaching. And when they come to research institutions, it's even worse. Because people in research institutions do not usually know how to teach. So graduate courses are very poor.
Okay, thank you, sir.
Yeah.
Sir, I have a question.
Yes, go ahead.
Sir, it was really a privilege to attend your lecture today. I learned a lot of things and your viewpoints. Sir, so you have been to places, you have conducted extensive research in various fields and subjects. And as you said, and as is clear from your lecture today, the world in itself is actually, the study of the world in itself is actually an amalgamation of chemistry, biology and various other scientific arenas. But sir, in the middle of all this, there is something we humans always ponder about, and there is the concept of God. So sir, personally, do you think there is an existence of God, or is it just us?
I shouldn't address my personal belief, but the idea of religion and the idea of God is a matter of faith. Faith and science are like two parallel lines which don't intersect at all. Faith is something that you have. Nobody can take it away from you. You have it. Science, on the other hand, requires evidence. Everything that I tell you as a scientist, you are free to disbelieve it. You are free to disbelieve it and provide experimental support to support whatever hypothesis you have. So science operates differently. So I don't think we should mix up science and religion. But if you think about these questions more and more deeply of chemistry and biology in detail, then of course the question of faith becomes irrelevant. You don't need any faith. You're still not understanding what all these things are doing, where did they come from. You're still looking. And you're quite happy. You sort of also accept many things. You accept that nobody's going to come to your help. Tell me, has faith helped anybody in the coronavirus pandemic? No. It has only helped people to be comforted in their mind. It has not helped in a practical way. Where are you looking for help in a practical way? You're looking for vaccine, no, coming from science. You're now clapping hands saying that in Oxford University or in Pfizer or in Moderna, they have now got the vaccine and hopefully we will also get it. So when you analyze, you're young, and when you analyze this connection between religion and science, it is best to keep them apart. There's no conflict between them, absolutely.
Sir, thanks a lot.
Any other questions?
Sir, I have one question.
Yeah, go ahead, Devashri.
Okay, so sir, I have one question. Is there any horizontal gene transfer from virus to human or vice versa? And if that happens, then does it make the virus more, I mean, survive in a better manner inside the human beings? Is that is happening or a proven thing is there? Experiment.
See, there is evidence for viral genes having been incorporated into the human genome. These are now what you would call fossil evidence for past interactions of human beings with viruses. So this has happened. In fact, I don't have it offhand, but Joshua Lederberg wrote about this in almost poetic terms about human beings and their interactions with viruses in the past. So that will happen. Our genome has a lot of history.
Yes. Thank you, sir.
Sir, I have a question.
Yeah.
Sir, if we see the primitive world, according to Oporin, Halden, the cause of a theory, everything, the first molecules were related to just the simple sugars and simple amino acids. Then higher organisms came and nucleic acids and lipid they came. Now the question is, sir, how the nucleic acid got attached to the only amino acid? Means DNA codes for protein, but it directly don't code for any sugar or lipid. Means how this correlation between amino acids and nucleic acid happen? Not with any other biomolecule, there is no correlation, but only between the direct correlation is between the nucleic acid and amino acid. Why is that, sir?
The short answer to your question is that I don't know and nobody knows. If you look at the literature, you will find there are many, many questions. How were the molecules of biochemistry actually chosen? How did the genetic code evolve? And would there be evidence of other ways of making proteins before this, the current way of making proteins came along? We don't know. The evolution of biochemistry is not really known. When you talk about Oparin and Haldane, they were speculating with what was known in their time. Today we know that we don't know.
Okay, sir.
Like, for example, people have speculated the genetic code appears to be optimized. And how did the genetic code really evolve? Were there other codes? People like Francis Crick and all have speculated on this even in the very early days. But much of it still remains in the realm of speculation. We don't have, unfortunately, a fossil record for the evolution of biochemistry. That means finding organisms which had slightly different biochemistry.
Thank you, sir.
So I think in interest of time, I think a lot of people would like to ask questions, but unfortunately the kind of interactions we can have in these COVID times is very, very difficult. And Professor Balaram, the common feeling is that it was a very stimulating talk, and it was a privilege to listen to you. But this, I think, is the beginning, and I think I am expecting you to spend time in RGCB, and I am going to pester you as I usually do, and I hope you will agree. And I am really looking forward to having you in RGCB during the time I spend here, and your guidance would be phenomenal for me to actually achieve something in RGCB, which has doing very well before even I took over. So thank you very much, and I'm really happy that you took a very short time to say yes to my request. And you have been always humble, and you have been always a very grounded scientist who actually tells what is correct, and you have been a very good orator. Thank you very much, and it's a privilege to have you here. And I think the audience would clap for you because you could hear the clap.
Thank you. Thank you. Thank you very much.
Thank you. Thank you so much, sir. And
Thank you.
we sign off.